This abstract is titled Diet Quality is Associated with Vaginal Alpha Diversity in the First Trimester of Pregnancy. The vaginal microbiome has been an area of earnest research interest, with hopes of manipulating an aberrant microbial community may translate into improved pregnancy outcomes. While some dietary components have been associated with improved vaginal microbial health, few studies have investigated the impact of comprehensive diet quality on this outcome. Our study aimed to associate diet quality as defined by the Healthy Eating Index, or referred to as HEI, with alpha diversity metrics longitudinally across pregnancy and healthy low-risk pregnant women. The study included 41 women who were recruited from the 4 largest ethnic groups in Hawaii. 10 from Native Hawaiian group, Filipino, Japanese, and non-Hispanic white were included. Each participant completed a food frequency questionnaire during each trimester, of which was performed at 12 weeks, 20 weeks, and 34 to 36 weeks gestational age. The Healthy Eating Index score was assigned from the food frequency questionnaire. Vaginal swabs were collected from the mid-vagina concomitantly at each time point, from which DNA extraction and 16S ribosomal RNA sequencing was performed. Alpha diversity profiles were assigned and linearly correlated with HEI score. Multivariate regression models were also used to account for covariates, including parity, age, obesity, and ethnicity. Results are demonstrated in the middle of the poster. Longitudinal data was available for 35 participants. 5 community state types, or CSTs, were observed in this cohort. Consistent with other descriptions of the vaginal microbiome during pregnancy, Lactobacillus species dominated for the majority of women. CST4 was primarily comprised of Epidemiology vaginae, Gardnerella vaginalis, and several other Prevotella species, along with anaerobic organisms. Figure 1 demonstrates the largest difference between the ethnic groups was of that non-Hispanic white participants and the rest of the cohort had lower amounts of CST3 or Lactobacillus inners compared to Japanese, Native Hawaiian, and Filipino participants. Table 1 demonstrates the Pearson correlation coefficient for alpha diversity metrics with HEI score for all samples collected. HEI score was correlated to chow and observed species number, but not evenness as measured by Shannon and Simpson indices. In the figure 2 panel on the left, the change in CSTs in each trimester are seen. CST3 decreased over time, while the rest of the CSTs increased. The right panel of figure 2 demonstrates the highest diet quality on the left, or first quartile, versus microbial composition of those who had the lowest diet quality, or the fourth quartile on the right. The main difference was an increase of CST5 in those in the fourth quartile of HEI score. Figure 3 shows the alpha diversity change of observed species number across trimester. This cohort demonstrated decreased alpha diversity in the 3rd trimester. And finally, figure 4 shows the primary contribution of this correlation of HEI and alpha diversity was from the 1st trimester with a statistically significant correlation coefficient of 0.48 R value. A linear mixed model did not identify any covariates that contributed to overall diversity except for trimester. In conclusion, our cohort describes the vaginal microbiome during pregnancy in a Pacific Islander and Asian cohort and was the first to do so during pregnancy. Lactobacillus species dominated in all participants across all trimesters, with Asian and Pacific Islanders having more Lactobacillus inners versus Lactobacillus crispatus and the non-Hispanic white group. Alpha diversity decreased throughout pregnancy in this cohort, and this trend is commensurate with other studies. Many factors influence microbial composition. In our linear regression models, HEI score was the only independent factor contributing to a dependent variable of observed species number besides trimester. Larger studies are needed to detect if higher periconception and first trimester diet quality impacts the vaginal microbiome and translates into better pregnancy outcomes. I would like to acknowledge that this project was funded by the Ola Hawaii project, which is supported by a grant from the National Institute on Minority Health and Health Disparities, as well as the Sharma Foundation.